like our clicker questions for today. So go ahead and get started. Alternative splicing and alternative tailing generally leads to different N-termini of proteins, C-termini of proteins, use of upstream ORFs, scanning A and B. I know there are more than 55 people here. That's more like it. Ten. 4. Okay, we seem to be pretty evenly divided between N-termini and C-termini, or just C-termini. So what is going on with alternative splicing and alternative tailing? What happens then? Okay, I guess it's daylight savings time and everyone's still not quite awake yet. So the idea here, okay, let's start out with alternative tailing. Alternative tailing, you cut your messenger RNA earlier than you otherwise would and add a poly A tail at that point. Okay, what does that mean about the N-terminus of your protein? It stays pretty much the same. C-terminus, on the other hand, is often very different. So what about alternative splicing? Basically the same way. One of the keys about alternative splicing is that usually it's only 1 or 2 exons that are going to be different in any of these alternative splicing scenarios, maybe as many as 3 or 4. But the vast majority of exons are going to be the exons which are always being used. And so that's why it's the C-terminus of proteins which change and not the N-termini. In most cases, the only time you'd actually get the N-terminus change would be when? If that alternative splicing or tailing happens all the way up in the 5' UTR, so untranslated region. Happens extremely rarely, although there are a few examples of how that has been known to happen. So our second question, the ADAR enzyme binds to single-stranded RNA, double-stranded RNA, double-stranded DNA. Single-stranded DNA is a ribozyme. So what does ADAR stand for? So adenine deamination acting on RNA. Exactly. So it's got to be A, B, or E. And it binds to one of the other ones. And if you were able to pull up that slide, you notice that it's a secondary structure which forms in RNA. And so that's going to be double-stranded RNA, which, of course, most of you got, which is wonderful. So you're all going to do extremely well next Tuesday morning at 8 a.m., right? Which is going to feel like 7 because it's daylight savings time. Yes, our brains will all be working at that point. Good. So those of you who have to leave, good luck, sayonara, et cetera, et cetera. It's dead week, so we're all being dead, right? Which actually is a pity, because I think that this is probably going to be one of the most exciting and fun lectures today. So you're all missing out, but your choice, not mine. So today we're going to talk about small RNAs, particularly regulatory RNAs, and some of the protein methods. On Wednesday, we'll talk about DNA methods, and then on Friday will be a review just of all the stuff we've covered since the last midterm. So small RNAs, basically, in terms of the small regulatory RNAs, really have to do with more double-stranded RNA, partly why I chose that question about the ADAR. You have double-stranded RNAs, which then lead to either cleavage of target RNA, translational repression, or even, in some cases, formation of heterochromatin. So, again, sort of ties together a lot of the stuff we've been talking about for the last couple of lectures and then earlier on in the course as well. Basically, this process has to do with the wonderful property of RNA, of course, is that it can base pair. And so you can get specificity of binding with an RNA sequence binding to either RNA, and usually it's RNA, but occasionally in DNA. But of course, in order to be able to do that, you've got to be able to separate those two strands, because if the two strands are together, of course, they can't interact with any other, at least not in terms of base pairing interactions with anything else. So you have processing that takes place, and then these are all together with proteins. We'll talk about these examples in just a second. Argonaut or Peewee proteins. These then will interact with a target RNA through base pairing interactions and then lead to some of these different options. Where does that double-stranded RNA come from? They're basically two sources, internal and external. And those internal ones are what are called the microRNAs or miRNAs. That's in just quick back-of-the-envelope calculation. These are usually on the order of a couple of hundred nucleotides long. They form double-stranded RNAs, like you have up here at the top. One microRNA can regulate hundreds of genes. And so this is one of the reasons that people didn't find them. And then when we talk about nucleic acid gels, the other reasons that people never found them is 100 nucleotides is about as long as what kind of really common RNA you find in the cell? Transfer RNAs. So transfer RNAs are about 100 nucleotides long. Anybody who's worked in a molecular biology lab knows you have this big blob at the bottom of your gel. It's all RNAs, so you throw it away. Well, that's where most of these things came from. They're mostly thrown away in the first place. So these are RNAs which have a specific sequence. They're genes. You can find these genes in the genome. Major way to detect them is that they've got huge amounts of self-complementarity. Yeah?
So did you just say that the microRNAs are genes that regulate genes?
So I said that microRNAs are genes. They are genes. There are genes for these microRNAs. So they're very specific genes. They're not a protein coding gene. They're not an RNA coding gene. Does that answer your question?
Kind of. So basically, they're genes that also regulate genes.
Right, so these are going to be genes which are going to be regulating genes. So these are gene regulatory genes, so not unlike enhancer binding protein genes or some of the other ones. We'll talk about the long non-coding. So yeah, genes which regulate genes, or I should say the product of the genes which then regulate the expression of other genes, to be more specific about it. So yeah, these, again, they're specific genes. You find them throughout the genome. Very often they overlap with other genes, particularly introns and exons, but it's not really critical here. But the main thing, again, is that they form these double-stranded RNAs, but that's because they're self-complementary to each other. These then get chopped into shorter pieces. Then these again get cut yet again. We're not going to talk about all the proteins that do this because you have to remember enough protein names already for this class. Once they've been cut to about 22, 23 double-stranded RNA bases, then they interact with some of these proteins which are important for the regulatory aspect of what these things do. Argonaut is one of the best known of those. Interacts with this double-stranded RNA, becomes single-stranded, and then there are two sort of mechanisms that can be used after this. If your microRNA base pairs extremely well, almost completely, with your messenger RNA that you're going to regulate, then there's an endonuclease activity which will cut in the messenger RNA. Once you've cut into messenger RNAs, we talked about with messenger RNA stability last time, you've got these free ends, a free 3' end with no poly A tail on it, a free 5' end with no cap on it. These are going to get chewed up really, really fast. And so the endonuclease comes from this Argonaut microRNA complex and chops everything up. That doesn't happen very often. It's actually relatively rare. Much more common is that you have a partial match between the sequences in your microRNA that you started with the 3' untranslated region of the messenger RNA which you're going to be regulating. This leads to a great reduction of translation. Okay, how is that supposed to work? Where does translation start? The 5' end. So what's this regulatory thing doing here at the 3' end? Okay, 3' and 5' end in messenger RNAs are almost always right next to each other. So that's one way that something interacting at the 3' end can be acting on the 5' end. The other thing is that when you have association of these microRNAs together with your messenger RNAs, that leads to these RNAs being transported to the so-called P bodies or processing bodies that we'll look at a little bit more in just a second. Yeah?
How is it that something with a more extensive match happens more rarely than a less extensive?
So the question is, how is it that you end up with fewer microRNAs that have very extensive matches and many more that have fewer matches? Well, if you just think about the number of nucleotides that you have there, how often are you going to have a stretch of 4 or 5 nucleotides that are going to match as opposed to a stretch of almost 20 nucleotides that match? Yeah?
When you say that the 3' end is often or almost always close to the 5' end of the mRNA, is that because of that circular?
Because of the looping, exactly. The reason that 5' and 3' ends are close to each other is because of the looping that takes place with the poly A binding protein, which interacts with EIF4G, that interacts with the cap binding complex. So that's where you've got the circular messenger RNAs. So that's what happens. Sorry.
And so is that specific to eukaryotes?
This particular mechanism is specific to eukaryotes, yes. We'll talk about a similar mechanism in bacteria in about 5 or 10 minutes. Okay, so that was internal RNAs, regulatory RNAs, the microRNAs. These are things that are all present, in genes, etc. Very similar mechanisms are used probably to deal with virus infection. So if you have an external double-stranded RNA, i.e. something which is not normally coded in the genome, and there are lots of good RNA viruses, or bad RNA viruses, probably depends on your point of view, which can infect, and it turns out also lots of eukaryotic cells get infected by these RNA viruses. They're single-stranded RNA when they're in the actual virus particle, but as we'll all learn about next term, at least those of you who are taking virology, RNA viruses always go through a double-stranded intermediate in order to make more of that RNA. So you end up with double-stranded RNA. So these double-stranded RNAs get processed. You know, that double-stranded piece ends up in a single-stranded piece together with the argonaute and other proteins. And then these RNAs, because they were made from the double-stranded RNA that came in with the virus in the first place, these are going to be almost identical to those sequences unless there's been some kind of mutation that's happened in them. And so what will happen here is that you'll have a perfect match between the RNA which has come in and the double-stranded RNA or the single-stranded RNA which is in the virus which comes inside the cell. So there you'll have lots of slicing, i.e. chopping up of this original RNA in the first place. So it's a really good way of regulating a virus infection. So great slicing up. Turns out that very similar mechanisms seem to be used to regulate the expression of transposons. So we talked about jumping genes again a few weeks ago now. These are particularly present in eukaryotic genomes as non-retroviral, reverse transcription-driven genes that are jumping around the genome, make lots of mutations because they're going to jump in pretty much at random. So you don't want to have too much activity of these genes. Turns out that you have many, many copies of these transposons. Those get transcribed. You'll have a messenger RNA that's transcribed one of these transposons. Another messenger RNA, as we talked about when we talked about the ENCODE project. There's lots of transcription going on in the genome all the time. You'll end up with double-stranded RNA for almost all the transposons in your genome. And because of that, and because of this mechanism that you use to regulate mRNA, and particularly mRNA stability, you'll be shutting down the activity of most of those transposons. So just presence of transposons, presence of this machinery is going to shut them down a lot. Centromeres are a slightly different story. Remember we talked about centromeres way, way back. Can't remember. That was back in the first midterm. I don't need to remember it because the final's not cumulative. Centromeres are these highly repeated sequences. Many, many copies of usually relatively short, again, about 100 nucleotides or so. But they're repeated sequences. So you've got repeated sequences. Anytime you have transcription across a repeated sequence, you're going to have transcription on one strand, transcription on the other strand. Now you're going to make double-stranded RNA. That double-stranded RNA will cause either degradation of those RNAs, but also that brings us to this second mechanism over here, which is RNA-induced transcriptional silencing. And so what happens here is you have a double-stranded RNA that forms on an RNA which is being made by the RNA polymerase, but instead of chopping this up, this leads to histone methylation, eventually DNA methylation, and silencing of that region by compacting the chromatin. So this is also what seems to be happening with places like centromeres, where you have constitutive heterochromatin, always compacted all of the time. So I mentioned P-bodies. What are P-bodies? Again, nice little name the cell biologists came up with for something they saw inside the cell. These are spots where if you have an antibody to, say, the argonaute protein, and you look inside the cell where that argonaute protein is, you find it in little spots. Here are a couple spots here. Here's a spot. There's a spot. Here's a spot. Here's a spot. Same thing is true with some of these RNAs. If you label some of the RNAs that you know are double-stranded RNAs, you find them in the same spot. These are the processing bodies where RNA is getting processed. And then when you stress a cell, you also find these same kinds of concentrations of proteins. They're not membrane bound. They're just concentrations of proteins and, in this case, RNA together in one part of the cell. And this seems to be important. That's where you're happening. All of this RNA processing is taking place. So the binding of Argonaut, the splitting of the two strands, the slicing, etc. All of this is happening in these P bodies and stress granules. So basically, that's just cartooned right here. You have your regular messenger RNA. It can be translated, or if there's some kind of regulatory element around, again, particularly if you've got interactions at the 3' UTR, that's going to be moved to one of these processing bodies where it'll get chopped up, it'll be maintained, etc. A few times, those RNAs will get moved to the stress granules. How do you know the difference between a P body and a stress granule? Slightly different proteins that are associated with each of these. Otherwise, if you look at them in the cell, you get little dots. That's about all you can tell. But stress granules, these are messenger RNAs which can get translated, whereas anything that's in a P body seems to be more or less destined for getting degraded. So P bodies, stress granules, and again, most of this is where that processing is going on. Mentioned this RNA-induced transcriptional silencing. Again, this is the cartoon of how that works here. Yeah?
Are the activities of the P bodies driven by trinucleotides, or is it pretty much energy neutral?
So the question is, energy in P bodies, is it mostly driven by nucleotide hydrolysis, or is it pretty much energy neutral? So I'll paraphrase your question and say, what about the dicing and slicing, those kinds of things which are happening? If you think about what's happening, once you've made a messenger RNA, that's entropically actually pretty unstable. So any kind of cleavage reaction is going to be energetically favorable. So you don't need any extra nucleotide hydrolysis at that point. So you will have to have, however, nucleotide hydrolysis in that first step where you're pulling the two strands apart. Because, RNA helicases, again, as we talked about for splicing, you've got to pull the strands apart. And pulling those two strands apart does require ATP hydrolysis, or NTP hydrolysis. Yeah?
Did you say that whether it's in a stress granule or a P body, it's more or less destined for degradation?
So it appears, and again, a lot of this is pretty new and hand-wavy. P granules seem to be non-active, and stress granules seem to be more active. So if anything, what seems to happen in the stress granules, you stress the cell. It's not unlike what we talked about last time with EIF2 being phosphorylated and then having trouble with recycling. It seems to be a way that the cell can sort of wait, hang out, maybe the virus infection's gone, then you can move forward and continue to translate, do all of your things. And so it seems a bit of a holding pattern. Hang on to those RNAs, make sure they don't get degraded, but they're not being translated at the same time. Yeah?
And did you say that the stress granules and the P bodies are always separate, or does a P body turn into a stress granule under certain conditions, or how does that work?
So the question is, what about P bodies and stress granules, and can they, co-transfer back and forth between each other? And so this gets back to what's the definition of a P body and what's the definition of a stress granule. And this is unfortunate that basically the way they're defined is by what proteins are associated with them. And so you look at, again, do some of these experiments like I showed you with the staining. You look at a protein which is specific to stress granules. You'll find it in one, in P bodies in the other. So it's hard to tell whether one is switching from one to the other. But basically, it appears to be, again, the presence of some proteins versus other proteins, which is the difference. And so you can't really tell whether one's switching or not. It's sort of a question that's sort of self-fulfilling prophecy. If the protein's there, then it is one of these, but that's the only way you detect it in the first place. So basically, you find two of these. P bodies you find all the time. Stress granules, not surprisingly, you're upregulated. You have many more of them under stressful conditions. Other questions about this? these small RNA processes. And again, this is the eukaryotic system. People thought, actually, that this system here, the CRISPR system, was originally something like an RNA interference system in bacteria, but they later found out that it wasn't. So we'll talk about that in a little bit more detail. So the idea here is that this is a virus defense mechanism that's present in bacteria. And it's, I think, a really cool story of how who cares about bacterial viruses, and it may turn into genome engineering. Very cool mechanism. But basically what's going on here is you have a DNA virus which infects a cell, and that DNA virus DNA matches some DNA which is present in the genome of that bacteria. If it matches, then there's cleavage of this viral DNA. So again, this is different because we were already talking about RNA before in terms of the RNA interference. Now we're talking about DNA cleavage that takes place. How that works, again, is a really kind of fun story. CRISPR is a really horrible name because it stands for, let's see if I can remember this properly, clusters of repeated intergenic sequences with palindromic repeats. Horrible. So just call it CRISPR. It's a lot easier to remember. Basically, what we have here are repeated sequences, and those are what's shown here in the orange pieces. So these are all repeated sequences. And lots of them, sometimes up to hundreds of these repeated sequences, all direct repeat sequences. And this is how they were originally found. They found all these interesting direct repeat sequences in the genomes of a bunch of bacteria, and it turns out archaea as well. And I said, oh, this is bizarre. What are these things doing? And at first people thought they were involved in chromosome partitioning, because bacteria have chromosomes as well. But then what they noticed was that in between each of these repeated sequences were these sequences, all different colors now, each of which, or not each of which, but many of which matched short sequences that were present in lots of viruses and or plasmids that had been found before. And so that brought people to the theory that, okay, this might be a way that the bacteria are going to be able to deal with some of these virus infections. One of the really fascinating things about this is it turns out that these spacer sequences, as they call them, the different colored ones here, vary based on infections by different viruses. And so this is a case of basically what looks like acquired immunity in bacteria, which people were like, whoa, what the heck? This isn't supposed to be working like this. But it clearly does. And there were some great experiments done, actually almost exactly 10 years ago now, actually less than that now. It was 2007 that the original papers were published looking at this. And it clearly is the case if you have one of these spacer sequences, you're resistant to virus infection. If you don't have one of these spacer sequences, you're not resistant to virus infection. So this opens up, of course, the big question is how you get these spacer sequences at all. So presumably a virus infection should be killing you off. How can you actually collect any of these DNA sequences? And about 2 weeks ago, there was a couple of publications in Nature about how people think that's happening. So this is a very new process which is going on here. How it works is it turns out that these sections of DNA that are present in the bacterial genome get transcribed, made into one big long RNA that gets chopped up into smaller pieces, and each of these smaller pieces, in a process very similar to what happens with Argonaut and all of the eukaryotic RNA interference machinery, these then interact with these RNA sequences and chop up the double-stranded DNA, which matches those spacer sequences. So this is really cool. It provides protection against bacterial virus infection. But it turns out if you put in that Cas protein and an RNA which matches a sequence in the genome that you want to work with that will lead to cleavage of that genome at that particular site. Now, why is this useful? Well, when you have a double-stranded break, how does it usually get repaired? Non-homologous end joining, which does what? Generates a mutation. What's the other way? Homologous recombination. So homologous recombination, you have to have a copy to give you homologous recombination with. So if you happen to have an extra piece of DNA, which you've put into the cell together with your Cas9 and your RNA, you can actually put in very specific DNA into any place in the genome that that RNA happens to match. So this is a really amazing genome engineering technology, which has literally just been developed in the last couple of years, probably going to be, at least a Nobel Prize. Some of you may have heard about, I forget what they're, creativity prizes, whatever. The founders of Google came up with these prizes, $3 million to scientists who come up with really cool stuff. That was, in fact, what they gave it to this last year, was genome editing using this. Who cares about bacterial viruses and bacterial virus infection? So this is the CRISPR, and this is something which you're going to be seeing and hearing much more about if you continue in the field, be it in a medical direction or in a research direction, potentially both here. It's a really fascinating process. And I'll upload a recent review article on this in D2L after class today. So the last part I wanted to talk about in terms of RNAs is also a relatively new field. These are the so-called long non-coding RNAs. We talked about long non-coding RNAs last time in terms of imprinting. Imprinting will lead to expression or not of some of these long non-coding RNAs. Basically, a long non-coding RNA is an RNA that's more than a couple hundred nucleotides long that seems to be expressed from a gene, probably regulatory, but we're not quite sure what it's doing. So, it's one of these sort of catch-all definitions. Long non-coding RNA, okay, when they're being made, what are they doing? Most of the time we have no clue. But in a couple of cases we do know what's going on. And most of those have to do with either binding to multiple different proteins, so serving as a scaffold. And we talked about scaffolds before. How do you bring multiple proteins together that need to be acting on the similar substrate, particularly when you look at RNA processing machinery, transcription. Most of these guys are all present in one place in the nucleus, in the cell, depending on what's actually happening there. So just bringing lots of things together. And so these long non-coding RNAs can be serving as scaffolds. They also, however, take advantage of the wonderful aspect of RNA. Why use RNA in the first place? Because it can base pair. And so that base pairing interaction seems to be able to either help out with binding to RNA. So if you've got some kind of RNA modification enzymes that are bound to your scaffold, you can bring them to a specific RNA, or you can bring them to a specific DNA through this base pairing interaction. Very often these will now be some kind of histone modification enzymes, so histone acetyltransferases, histone methyltransferases, which you'll then be able to bring to the DNA and cause changes to take place to it. So these are just a couple of the examples. There are literally thousands of these long non-coding RNAs that are present in our genome. They seem to be relatively well conserved. You find them in mouse genomes. You find them in other genomes as well. What exactly they're doing, we only know about probably a handful of different cases there. But they're probably important for many other things. It's just a case of keeping us employed for a while. Okay, any questions about RNA and regulation? So now we're going to talk about methods, which in some cases I think we should talk about methods right at the very beginning of the course rather than at this point in the course, but the way our textbook is set up, the way most people teach it, do methods towards the end. Those of you who are unlucky enough to take this course, the next time I teach it, we may do it a different way around. Yeah?
You were talking about it's relatively conserved,
Mm-hmm.
and then you gave some examples of eukaryotic
Okay, so the question is, do you find long non-coding RNAs in bacteria and archaea as well? The answer is yes, you do. Do we know what they're doing? We don't have a clue. No. And there seem to be many fewer of them. And so if you look at most, and we talked about this, again, way back when, like 9 weeks ago. Can't remember any of that. If you look at genome sizes, most bacteria and archaea have pretty compact genomes, whereas many eukaryotes have ridiculously ginormous genomes. So you've got a lot more space to encode some more of these longer RNA sequences. So you definitely do have small RNAs in some bacteria and archaea, but some of these longer non-coding ones you seem to have fewer of. But they're definitely there, and the question is, what are they doing? Or are they just random transcription, which seems to happen quite a lot, which is strange. You wouldn't think that's kickies, but it does. So that's one of the open questions. So yeah, in terms of methods, sorry, any more questions about the RNAs before we move on and talk about methods? Yeah.
When you're talking about the proteins that associate with microRNAs and the small interfering RNAs,
Mm-hmm.
are those attracted to those RNA targets because of the secondary structure? Is that what is that why those proteins can associate with both internal RNA and RNA from an external source?
So the question basically is, where do you get the specificity from? I'm paraphrasing. Where do you get the specificity for binding of things like Argonaut and the RITS complex, et cetera? Most of that seems to come from just the fact that you've got these relatively long, this is 20-plus nucleotide double-stranded RNA. And that's, if you think about it, it's considerably longer than what you see in tRNAs. The stems of each of the tRNAs are considerably shorter than that. And so it seems that 20-some nucleotides, and usually actually they're longer than that. They're about 30 nucleotides that are double-stranded. You don't seem to find them in other cases of completely double-stranded or very nearly double-stranded. So it seems to be mostly that linear double-stranded RNA which is most important for getting the binding in the first place. So it's not a sequence-specific, because if it were sequence-specific, these things couldn't work in general. It just seems to be that the fact that you've got this double-stranded RNA. There's probably actually selection against, evolutionarily speaking, other RNAs that'll base pair like that because they'll get degraded. Okay, other questions? Yeah, so in terms of methods, there are basically two different sets of methods. We're not really going to talk much about cell methods here, mostly proteins. And then again, on Wednesday, we'll talk about nucleic acid methods, including sequencing. Why should we care? You can read that. Basically, everything we've talked about has to do with this, which is why I've been thinking about maybe doing this earlier on in the course rather than right now. So what are our basic concepts here that we're going to talk about for the next half hour or so? Really dealing with proteins, and mostly how do you separate them from each other? If you think about a cell, it's got a whole mixture of different proteins in it in various different amounts. And so most of the proteins we've talked about for the last couple of lectures have been those gene regulatory proteins that are present at very small amounts, but lots and lots of different ones. And so actually finding those particular proteins is really hard. And that's what a lot of these techniques are about, is how you actually go about finding these particular proteins. And usually that has to do with separating them from all of the other proteins which are there and identifying them. So basically, this first part here is all about separating them, and the last part here is about identifying them. Broken the outline down a little bit more, talking about centrifugation, there's really sort of three different kinds of centrifugation. Either a preparative method, which is basically really big, or analytical methods, which is relatively small. These are sedimentation and equilibrium centrifugation. We'll look at examples for all of these in just a second. Chromatography. How many of you have done chromatography in your chemistry lab? Separate different colors. This is basically exactly the same thing, only we're not separating colors or dyes. We're separating proteins, and those separating the proteins based on their different structures. That'll either be the charge, the size, or whatever they're interacting with. So that's basically all of our purification techniques that we'll be looking at. Analysis, electrophoresis, which we've talked about a little bit already, particularly for proteins, SDS PAGE, so SDS, sodium dodecyl sulfate, PAGE, polyacrylamide gel electrophoresis. Yes, we didn't have enough acronyms already. And then a little bit about what are called 2D or two-dimensional gels, which is a higher resolution way of separating proteins from each other. But this is in an analytical fashion. This is very rarely used in a preparative fashion. These days, almost everyone's doing mass spectrometry. Mass spectrometers have gotten a lot cheaper, a lot better, and we have a whole bunch of genome sequences. And so it turns out that mass spectrometry is really sort of the method of choice for identifying some new protein, which you've just isolated through the processes which we have over here. Once you've identified that, usually you want to know what the structure of that particular, my favorite protein is. Two main ways that people get its structures these days are X-ray crystallography and NMR spectroscopy. These are really hard, time-consuming techniques. Very often, the structure of one protein will be a whole PhD thesis. So gives you kind of an idea of how difficult these processes are. And that leads us to what most people end up doing instead of this rather complicated process, X-ray crystallography and NMR spectroscopy, which give you wonderful structures and huge amounts of information there because your structure tells you something about function. The bioinformatic process is basically taking advantage of the fact that people have done all this work already and comparing what you have to what other people have already found. And if you're lucky, then people have done all the really hard work and you can come in and collect all the spoils there. And if we get to it, we'll talk about protein-protein interactions today. Probably end up talking about those again on Wednesday. Let's start out with centrifugation. And part of the reason to start out with centrifugation, this is usually one of the very first steps that you do when you're trying to separate your favorite protein. And again, we'll say your favorite protein happens to be some kind of transcriptional regulator because those are the coolest proteins by far. So you start out with your cells or your tissue or whatever it is, and you break it apart into lots of little pieces. And then what you want to do is separate each of those pieces from themselves. One of the easiest ways to do this is through centrifugation, just spinning. And this is really nice because it can use literally up to liters worth of material. Again, back in the late dark ages when I was working at a biotech company, one of my colleagues came in with a horse pancreas. And that's what we started with in terms of trying to purify particular proteins from that. So not unusual. We took it, we ground it up, we put it into the centrifuge, and basically centrifugation is great because it separates based on pretty much the size, to some extent the density of what you're looking for, particularly in these preparative purposes. So you take your ground up horse pancreas, you put it in your centrifuge, spin it for a little while at relatively low speed. Most of the junk comes down here at the bottom, and you usually throw this away. This is all of your extracellular matrix, all the lipids, etc. So this is great because now you just take what's present up on the top after you spun this. Now you spin this a lot faster, a lot longer, and now you can start to separate out things like mitochondria, etc. You keep spinning faster and faster and longer. Eventually, you can end up with ribosomes, viruses, etc., the really big stuff down here. And usually, what you care about is what's up here, because the proteins that you're looking for are usually much smaller and more soluble than everything else. But you need to get rid of all of this extra stuff in the first place. And so centrifugation is a great way to do this. Large scale, relatively straightforward to do this. The only major issue is particularly when you're doing some of these really high-speed spins. These things are going about 100,000 RPM, 100,000 RPM piece of a couple pounds of titanium. You want to get away from it because these skins are really pretty scary. And one of the things that you notice on here, hopefully, is the armored chamber. If you want to see some really scary things, go on YouTube. Look for broken ultracentrifuges because they're pretty scary. But it's a great technique. We use it all the time for first pass for purification. So the other kind of centrifugation that you can use is much more of an analytical technique. And this is basically trying to get some idea of your protein, your, in some cases, nucleic acid, or even your subcellular organelle in terms of its size or its density. So two different ways of doing this. Sedimentation velocity, which is over here on the right-hand side, sorry, left-hand side, my right, your left. So left-hand side, and then the right-hand side, which is equilibrium centrifugation. Here, you put your sample at the top, you spin it, and basically, as you're spinning this, your sample, which is a mixture up here at the top, will separate based on how fast it goes through this gradient. Now, once you're done, you've spun this for a certain period of time, then you make a little hole in the bottom of your tube, and you just collect from the bottom towards the top. And so this tube over here represents what came out of the bottom right here. The next tube, what came out next, and so on and so forth. Just across each of these individual, people will call it a fraction. So just basically as if you were taking slices off the bottom of this tube. Here is what's gone relatively quickly through your sedimentation velocity, and here what's gone relatively slowly. This is fast sedimenting. This is slow sedimenting. Everybody remember your ribosome components, 16S, 18S, 30S, 70S. All of that comes from exactly this kind of experiment over here. It's these, sedimentation. So this gives you a really nice idea of what size your particle is that you're looking at. On the other hand, if you want to know what the density of your particle is, you want to use this equilibrium centrifugation over here on the side. Where did we first see equilibrium centrifugation? Measles and Stahl, exactly. So how was originally shown that DNA replication was semi-conservative, was looking at differential density. Now, what did Measles and Stahl do? They looked at N15 versus N14 DNA. Very small changes that you have between these two things, but you could separate them relative to each other. So this equilibrium centrifugation is an incredibly high-resolution technique. You can separate things that are extremely similar to each other through this process. What happens is you end up setting up a gradient, in this case cesium chloride, so a heavy metal solution. That sets up a gradient really fast. When you turn on the centrifuge, you'll have dense down here at the bottom, less dense up here at the top. And then whatever you put in there, say it's your DNA, will now, in this centrifugal force, again, these are usually spun at tens, if not hundreds of thousands of RPM, which is hundreds of thousands of G, they'll be pushed through centrifugal force to the place where their density is now equal to the density of the gradient on the outside. And what they end up with are these bands, so a low density band and a high density band. And so this is exactly what Measles and Stahl did. They saw that you had these two different bands of different densities of mu and old DNA. This is great, again, analytically. Turns out you can use this equilibrium technique for separation as well. We use it for viruses, but it's a really low throughput technique. Much better technique for getting purification is column chromatography, and this actually is shown here in the figure as, again, separating things of different colors. But basically, it's the same process. Now we're just looking at different proteins. All that we have is a column. You put your sample at the top of it. Depending on what's here in this gray stuff, some things will go through here quickly, some things will go through here slowly. So all that's happening here, it's the matrix, which is here, the gray stuff, which will slow things down. Ion exchange, hydrophobic interaction, gel filtration, and affinity. And we'll take a look at that as we move along. You also just collect fractions off the bottom here, just like you did with the centrifugation. And what is least well bound comes off first. What's more bound comes off second. Unfortunately, most proteins don't have these pretty colors, so you need to have some way of determining where your favorite protein is. For instance, an electrophoretic mobility shift assay to see where it's looking at. So let's look at some of these matrices. Also, people will talk about these as resins. Talk about three. There are a few more. Ion exchange, gel filtration, and affinity. So ion exchange is you have your matrix, again, that's here in gray, that has a specific charge on it, and that charge is very stable. So here we have a matrix with positive charge. You put your sample on this. Anything which is negatively charged will interact with this positive charge and it'll get slowed down. So something that has positive charge will go through your matrix very quickly. That'll be in your first fraction. Anything with a negative charge will bind to it. And then, in order to get that protein, usually, which is bound to the column off, you just increase the salt concentrations. And so that's the exchange part of this ion exchange. So put in salt. That'll compete with your protein, and your protein will come off. We'll see an example of that in just a second. Another one of the most commonly used here is called gel filtration or size exclusion. Again, here you have a matrix in gray with small holes in it. And these holes are very tightly defined in such a way that small things get into these holes and are slowed down. Big things have a more harder time getting into the holes. They're not slowed down as much. So big things come off first. Small things come off later. This works pretty well. You get about 20 times purification. So that means if you had 5% of something in your sample that you started with, you could end up with 100% of it at the end. Much more likely you've got 0.5% and you end up with 10% of it at the end. So this is great, but it means that usually if you're talking about your 0.1% of protein that you have, which is your transcriptional regulatory protein, you have to go through lots of these processes in order to get it. If you have a way of specifically binding to your favorite protein, this is a much better way to go if, however, you happen to know what it's binding to. We already talked about the DNA affinity chromatography before. It's how you have a specific DNA sequence that your protein binds to. That's only one way. If you have antibodies to your favorite protein, then you can use antibodies to bind to these matrices. You know something else that your protein binds to. You can bind it to these matrices. Or what most people do these days is they have made their favorite protein with a little tag on the end of it. And that tag is almost always going to be used for purification purposes. And a lot of these tags will be either 6 histidines. These tags, by the way, are put on with recombinant DNA techniques that, again, we'll talk about on Wednesday. Histidine tag, what's called a flag tag, maltose binding protein, glutathione transferase. All of these things are something you hook up to the end of your protein in order to make it easy to purify. And again, using the recombinant DNA technique. So now what you have is something that binds to the tag, which is on your beads, and allows you to get really good purification through this process. So let's look at a couple of examples. These are actually kind of fake examples, but be that as it may, it's what we're looking at. So up the top here is a typical, quote-unquote, ion exchange chromatography run. Look at the red line. That just represents all of the proteins which you have in here. You put your sample here on the top of the column. You wait for a while. These will be all the proteins that don't bind. which will be those that have the same charge as whatever's on your ion exchange column. And then you slowly increase the salt concentration. That's this green line. And at a certain salt concentration, you'll see activity, what you're looking for. You'll notice there are a whole bunch of peaks here. So this is a whole bunch of different proteins that you have here. And there's still probably quite a few proteins here in the peak that you started with. So now you take this peak and put it onto a size exclusion or a gel filtration column. This will also separate, now again, based on size. The larger ones come out at the beginning, and the smaller ones come out later. Here again, you have your activity. Again, let's just say it's a DNA binding protein. This is your electrophoretic mobility shift assay. All the proteins that are present in this fraction have that activity. A couple things about gel filtration as opposed to your ion exchange. It's a relatively low resolution technique. It doesn't purify as well, but most importantly, it dilutes. You see that you've got some nice, relatively sharp peaks here. That means that you have a high concentration in one of these fractions. Here they all get spread out, and that's because all that you're doing is slowing down the process of these proteins through your column just by them interacting with the beads. So again, the best way, if you have a way of doing this, is your affinity chromatography, where you put your fraction onto the column. Everything that doesn't bind to your ligand flows through, and these bars here are supposed to mean that this is almost all of your protein. And then you have a little peak here at the back end, which is where you actually have what you're looking for, and hopefully it's only the one protein that you're looking for. And in SDS page, hopefully you'll end up with one band over here. We'll talk about SDS page in just a second here. Yeah?
Could this also be a way of sort of fishing for something that you think could be like a protein that you're familiar with, like a new, I don't know, because you need to know in advance.
Right. So the question is, could you use this as kind of a fishing expedition? Can you actually try and find something which binds to your favorite DNA sequence, for instance? You certainly can. Usually what you'll find is the amount which is there is often very, very small. And usually you will have known what that protein is to start with. And so, but this process, again, I'll tell you a little bit more of my story here. When I was working for this biotechnology company, when we had the horse pancreas, we were trying to find a protein that bound to a particular drug that the company was making. And so we put the drug on the column. And then we tried to find a peak like this. And it turns out, yeah, there actually was something in the horse pancreas which bound there and came off of it. So you certainly can use this kind of technique for doing that. As I mentioned before, most purification these days has to do with tags, also known as fusion proteins, where you have the gene for the protein that you're looking for, that you may have found in many different ways, and then you add a little piece to the end of it, which makes purification much, much, much easier. Sometimes it works, sometimes it doesn't, but of course you have to have the gene for that protein to start with. And that's, usually you've had to have purified the protein in order to get the gene in the first place. So it's great if you know what you're looking for and made some little changes, but the original one, usually you've got to go through all these other processes in order to find it. So SDS page, which I promised to tell you, this is SDS page is a particular kind of gel electrophoresis. Here, it's just a very general gel electrophoresis. You have a cathode, which is negatively charged, an anode, which is positively charged, some kind of matrix between the anode and the cathode, which will slow down whatever you've put here on the top. So it's not unlike your column chromatography here. This matrix will slow things down. These things are migrating in an electric field. In a standard gel electrophoresis, you're separating based on charge, because that's going to give you the mobility and the size. That usually is confusing because you end up with multiple different things. It's much better to have something which will just separate either based on charge or based on size. And size is usually, well, what we care about? No, let's not get into that. But the proteins separation based on relative molecular mass. It's usually get much better separations this way, and you can sort of say, oh, this is a particular size of a particular protein. The problem is, and what you've actually used to do the purification in the first place of using ion exchange, is that proteins have very different surface charges on them. So you need to change that somehow. Well, the way to do that is to treat them with detergent, which will denature your protein. Now, here's a particular detergent, and this is one of the most favorite ones here, sodium dodecyl sulfate. Typical detergent has part of the detergent which is highly hydrophobic, part which is hydrophilic. You mix this together with your protein as long as you've heated it up. So you heat up your protein comes apart. Then this detergent binds to it and gives it a uniform charge density. So the charge is basically the same due to this detergent relative to your size. You also need to break disulfide bonds, so you include beta-mercaptoethanol here. So that's shown here in cartoon form. Here's your protein bound with a disulfide bond. You heat it up with SDS and mercaptoethanol. It gets this uniform charge density, and now these things can be separated in a polyacrylamide gel. So we'll just look at one example here. Here's my purification process up here at the top. Here's our broken cell. This is what we started with. Then we do centrifugation. You'll notice many fewer proteins that you have here. Now we've done our ion exchange chromatography. We're getting down to fewer and fewer proteins here. We do our gel filtration, this one, and then finally our affinity chromatography. And we have just one band at one particular molecular mass, and this is hopefully our favorite protein. We've been going through the process, of course, the whole time. Each of these fractions we look at with our, again, assuming it's a DNA binding protein, electrophoretic mobility shift assay, and you can see that this one particular protein is the one which has the activity that you're looking for. So this is great for separating based on size. You can also separate based on charge, and that's what's called isoelectric focusing. Here, again, you take advantage of the fact that your proteins have different charges on them. These charges, of course, are going to be dependent on the pH of whatever solution it is that your proteins happen to be in. It's the pKa of the individual amino acid side chains. So your proteins will have a positive charge if they're below the pKa, a negative charge if they're above the pKa, and if you have a gradient in pH and just put your proteins into it, let them sit there for a while, they will end up at a position where they have no net charge as long as they're in an electric field. And this is, in fact, an example of one of these what they call isoelectric focusing cells up here at the top. Very high electric field. These will migrate until they end up at a position where they have no net charge. The main problem that happens here is when they have no net charge, they also don't interact with water very well, and so they very often will precipitate and fall out of solution right when they're at this what's called the isoelectric point here. It's a very high resolution way of doing separations, and you can combine it with SDS Page on 2D gel electrophoresis, which we'll talk about on Wednesday.